you have to put distilled water or buffered water. Now, what is available here is buffered water. If it is distilled water, it is double the number of drops of the Leishman stain. So, as you have to count this to make the quantity correct, whatever is required for proper staining. So, first you have count the Leishman stain number of drops, otherwise how many drops you will put the buffered distilled water or buffered water. If it is distilled water, it is double the number of drops. For buffered water, it is usually said that it is the same number of drops as the Leishman stain. Now, after putting this, so after putting the Leishman stain, wait for 2 minutes, then 1.5 to 2 minutes. After putting the buffered water, again you have to wait for 8 to 10 minutes. That is called the staining time. Then what happens is, whatever it is called, something called the greenish scum, whatever lies the fluid over the slide, it is called the greenish scum. Scum literally means dirty. So, greenish scum you have to wash it off. So, for washing it off, it has to be put under, it should be done with tap water. Ideally, it should be done with tap water. Now, what happens if you allow tap water directly to fall over the smear itself may get washed away. So, the rule is just gently switch on the tap gently, so that water is not flowing at a very fast rate. Very slowly water is flowing, and wherever you had taken your blood drop, on that area put your thumb like this, and you are allowing the tap water to fall on your thumb very gently, and very gently it will wash away the whatever scum is over the fluid, and the smear has got fixed. Smear gets fixed in the 2 minutes, in the first few initial minutes, and we wait for 2 minutes more after putting the stain, and the smear has been fixed to the slide. But fixed means it is, that means if so fluid is flowing at a high rate over that smear, then it may get washed away. But fixing means this, scientifically it is called as fixing. So, this fixation of the smear, proper staining is after putting the buffered water. Okay, then you wash away the greenish scum, and then after drying all this, after washing away the greenish scum, after drying the slide properly, we can see it under the microscope. Now, how to identify the cells under the microscope? Most of you know by now. For those who are new, I will go to the to your table and help you identify. Okay. So, now start. Today it is 12:10, okay, roughly it is 1 hour. In the exam you will get 45 minutes for this topic to complete, and then there will be, the examiner will come and ask you questions. Whatever experiment is there are 7 or 8. So, some ones are short, takes roughly say 10-15 minutes, they can be completed. So, there can be more than one experiment if someone is allotted, the question is allotting shorter experiments. For differential leukocyte count, you will get 45 minutes. It is almost 1 hour, but the official time allotted is 45 minutes. This is the final destination, and if you count under the, you know about the objective lenses, when you count under the 100x lens, oil should be used, and the position of the condenser, this one, this position of the condenser should be the highest position. Whenever we do some experiments where this 10x lens is used, objective lens, in the condenser position is the less position. This is the eyepiece there. So, these are which will be practically required, but you have to know the parts of the microscope, how to hold the microscope. How to hold the microscope? Do you know? Yes, you. How to hold the microscope? Suppose you are carrying the microscope from one place to the other. 100